Hello, everyone, and welcome to today's live broadcast, evaluating the kidney chip for transporter-mediated drug-drug interactions applications. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Emulet. For more information about our sponsor, please visit their website at emuletbio.com. So let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Dr. Stephen Anderson, Senior Vice President at LabCorp Corporation of America Holdings and Chief Scientific Officer at Covance, Dr. Kyung-Jin Jang, Vice President of Technology Implementation and Field Science at Emulet, and Dr. Donald McKenzie, Vice President, Head of Global Metabolism. For a complete biography on all of our presenters, please visit the Biography tab at the top of your screen. Dr. Anderson, you may now begin your presentation.
Thank you for that introduction. Hi, I'm Steve Anderson. I'm the Chief Scientific Officer for Covance, and welcome to our webinar this afternoon. I'm joined by colleagues from Emulate and Covance for us to review with you a very collaborative project advancing organ-on-a-chip technology to look at applications for drug-drug interactions, transporter-mediated drug-drug interactions with a kidney chip. So what we'll be reviewing today I'll give an overview of the need for better preclinical models in the area of drug development. We'll focus on that with regard to a renal chip. I'll hand it over to our colleague KJ to talk about the development of a proximal tubule kidney chip on the Emulate platform. And then she'll hand it over to Don to talk about our assessment of the functionality of that renal chip, looking at morphology, expression of key biomarkers and transporters, the assessment of the function of those transporters, and then evaluation of drug-drug interactions. So for preclinical models, which are essential in the drug development process, they provide a variety of challenges. One of the challenges is the predictability of those models and the lack of predictability, which leads to a significant attrition during the drug development process. Those models have gaps in the ability to address safety concerns of new molecules, as well as mechanism of action and potential efficacy. The assessment of safety or toxicity of a new asset in preclinical models is impacted by a variety of factors. The complexity, the fact that there are multiple contributing factors, the role of multiple cell types in the expression of that toxicity, and very critically, the fact that there are species differences as you try to translate data from an animal species into what you might expect in humans. One attempt to try to bridge that gap is to look at other preclinical or non-clinical models and cell-based models. These include in vitro models and also in silico models that take computer assessment of potential interactions that would occur within humans to try to understand and have better predictability. Animal models, which are really the staple in drug development, have a number of challenges and features that are relevant in the process. One, as we've talked about, is the translatability of data from animals into humans due to species differences. Another is overall cost. So there's great interest in vitro models, human cell-based models, to try to address that. Variety of potential uses. Again, in candidate molecule screening, that's been something that's been used for a long time. To do mechanism of action studies, and then also to look at that translation from preclinical into clinical. The challenges that still exist in these systems is there a physiological equivalence from a cell-based model to an organism expression of that toxicity or that parameter. The other is we're dealing with single cells most often, so can we translate this into a whole system? So again, they also provide challenges. We'll try to address some of those today, but there are a variety of cell-based models that have been utilized in drug development. Everything from traditional two-dimensional cell cultures, 3D cell cultures, bioprinted cells, organoids. Today we'll talk about the development of an organ on a chip modality, multiple organs on a chip, and even in the cancer space, tumor spheroids, to try to really understand things in cancer like tumor heterogeneity. The potential advantages of these systems are reduction in cost, potential efficacy in the timing of that assessment, and then the decreased use of animal models in the preclinical space. There are opportunities to improve these models. One is the incorporation of biomarkers. You'll see a little bit of that today. The ability to leverage or combine models, and then improvements in models. And today we'll focus on some of those improvements, leveraging the organ on a chip technology. There's a number of applications that really are in front of us today and on the horizon for such models. First of all is the optimization of candidate selection, again, being a more holistic look. We are always interested in reducing the dependence on animal models and redistributing those resources elsewhere so they fit well within the 3R approaches that are very much of focus within the industry. There are applications potentially where systems are currently inadequate. The model that we'll talk about today fits very well into that. And then the opportunity to integrate both pharmacological and toxicology studies, again, providing some of examples of that in the discussion today. The relevance for an advanced model in kidney or renal functionality is highlighted in this slide. There's a number of interesting publication and guidance around assessing drug-drug interactions. And in the middle, the gray two bubbles highlight some of the current gaps that exist today. We'll talk about those in greater detail as we review the development of this proximal tubule kidney chip and its application. But many of those include the ability to translate that in vitro model, that in vitro system, into something that allows to look at drugs transporter and drug-drug interaction. And then also the ability to take that data and model it in a way that makes sense as you move from an in vitro model into an in vivo situation. So with that, I'm going to turn it over to KJ, who will start to talk and provide details on the development of the organ on a chip technology. So KJ, over to you.
Thank you so much, Steve, for the great overview. So from here, I will highlight how organs and chips can recreate the complex cellular microenvironment by combining different key components. So within the organ chip, we can build tissue-tissue interface by establishing epithelium and endothelium layers on each channel. And the porous membrane, which can be coated with extracellular matrix, allows the cells to communicate. A key feature of our organ chip is the ability to create dynamic environment by applying different degrees of flow or mechanical forces. And not only that, we can build additional complexity by adding circulating immune cells within flow or a stromal compartment or interstitium by utilizing ECM layer and stromal cells inside. We can also add pathogenic or commensal microbes on the tissue layer, and we can even create hypoxic system to provide more in vivo relevant microenvironment within the chip. So all of this is designed to build organ-on chips to be used for various applications, including compound efficacy, safety, pharmacokinetics, and mechanistic investigation. First, let's take a look how we can build a human proximal tubule in our chip. Immune-led organ chips are made of flexible, transparent polymer that has two independent microfluidic channels separated by a porous membrane. We can culture the proximal tubular epithelial cells in the top channel and renal endothelial cells in the bottom channel. Then we can apply fluid flow independently in both channels to create a dynamic environment like within our kidney. So the porous membrane allowing molecular transport and fluid flow in both channels are critical components to recreate physiologically relevant renal environment in our chip. And to give you a sense of scale, the surface of the cell culture area in the chip is approximately 70% of one well of the 96 well plate. Now, let's take a look how we apply flow within our chip and pod. So after establishing the tissue culture within the chips, they're connected to this unit called a pod. The pod contains inlet and outlet reservoirs for both top and bottom channels. Once we start the flow in our automated culture instrument, the media from the pod inlet reservoirs enters the respective channel in the chip, travels across the channel, and then collects in the outlet reservoir. And we are not showing our instrument here, but it is a pressure-driven unidirectional flow system without any tubings and syringes. Then these schematic views show how the volume changes from inlet to outlet over time. You can add up to 4.5 ml of culture or dosing media in each inlet when you start. Then you can set a flow rate from 0 to 1,000 microliter per hour in our instrument. So at time 2 on the right, you have increased volume in the outlet reservoirs over time. So for the kidney chip, we apply 60 microliter per hour of flow rate. And the effluent sample can be collected at any time during the culture. If you collect daily, you have around 1.4 ml of effluent from each channel for the kidney chip. And you can run different types of biochemical analysis or measure parent compound or metabolite concentration directly from this effluent. Also, cells from each channel can be harvested, lysed, or stained directly in the chip once the experiment is finished. And you will see examples of transcriptomics and staining data later. Then let's take a closer look inside of the chip when we add dosing solution through the pod. So here we can add the compound either to the top or bottom channel to run a top to bottom or bottom to top type of transport study across the cell layers under flow. Also, multiple compounds can be added sequentially or simultaneously based on your experimental design. Also, not only small molecules, but for example, biologics, viruses, immune cells, or other types of components can be added in a similar way. And you can measure not only a transport across the cell layer, but also complex cellular interaction or responses from the cells in the chip. For the drug transporter study, we first checked non-specific binding or absorption level of the compounds to the chip material. As you can see, the key points that we summarize on the top right, compounds that are substrates of active transport in the kidney tend to be hydrophilic, which generally corresponds to minimum compound loss due to silicon-based material absorption. And this is shown in the reference on the left regarding the extended clearance classification system. So when we tested the material absorption of the probe substrates that we use for the drug transport studies, as you can see on the bottom right, recovered compound concentration in the chip effluent, which were normalized by the control dosing media, showed no absorption for all substrates into the chip material as we expected. Had any compound shown a tendency of absorption, we have a means to measure it and proactively understand and adjust for any potential impact through a simple kit specifically designed for this purpose. Now let's take a look at a specific example of the impact of flow. This is the data from rat collecting the kidney chip, a different region of the nephron that I studied 10 years ago while developing these organ chips. So under static condition, you can see dense actin fibers covering the entire cytoplasm. However, once we start the flow, you see a significant change within just a few hours, rearrangement of the cytoskeletal structure, especially accumulation of actin filaments at peripheral cell borders. And there are other similar reports on the proximal tubule cells as well. So this structural change and flow further contribute to correct localization of transporters to apical or basolateral membranes. And this example clearly demonstrates the importance of flow for maintaining a more physiologically relevant renal culture system in vitro. Then let's take a look how Emulate team has optimized the human proximal tubule kidney chip as part of our collaboration with Covance. The proximal tubule is where glomerular filtrate first pass through, and this is highly specialized region for various molecules reabsorption and secretion between the tubular lumen and peritubular capillary. So for the model optimization, generally, critical components that we focus are good quality of the cell, tissue-relevant ECM, proper seeding density, and order between epi and endo, media, and flow rate. And you can see on the right table some key information from the snapshot of our protocol after intensive optimization work with broader ranges of experimental parameters for each component. And we have a full protocol of this model available. One note regarding the stretch, while this is beneficial to improve functionality of some of organ models, including alveolus, intestine, or glomerulus, this was not necessary for the proximal tubule based on our comparison study earlier. So we only apply flow, not stretch for this model. And we have published it on a glomerulus chip model as well. Here are some example images of the characteristic morphology of the proximal tubular epithelial and renal endothelial cells within the proximal tubule kidney chip. You can see monolayers of both cells maintained throughout the culture underflow in the chip. And you can monitor this type of cell morphology under the bright field microscope anytime during your culture. And these both cells are kidney-specific human primary cells, especially co-culture with renal endothelial cells is important. In the kidney, both glomerular and peritubular capillary endos are fenestrated, allowing molecular transport. And the one that we are using, also commercially available sources of renal endothelial cells are from glomerulus. We can also stain and image specific markers of both epithelial and endothelial cells directly in the chip using standard fluorescent microscopes, including confocal and high-content imager. We can label the primary cilia by immunofluorescence staining of acetylated tubulin or a scanning electron microscope. And from that, you can also see brush borders in the proximal tubular epithelial cells. We also checked other markers, including adherence junction protein, beta-catenin, and tight junction protein, occludin, water channel protein, aquaporin-1, and sodium potassium pump, all indicating correct localization of these markers. And for the renal endothelial cells in the bottom channel, we confirmed the expression of the key endothelial cell markers, including CD31, PKM1, and VHE-cadherin. So from all of the characterization data and numerous studies that we have performed within the proximal tubule kidney chip, we are confident that we have established a viable, functional, and dynamic human kidney model in our chip. So with that, I will hand over the presentation to Don to provide details on the transporters and DDI studies using this chip. Thank you.
Great. Thank you, KJ. Really appreciate it. Again, thank you, everybody, for taking the time out of your day to attend the webinar. So when we entered into the collaboration with Emulate, one of the goals that we had was to establish the list of requirements that are going to be needed to generate a translatable model to fill that gap that Steve mentioned towards the beginning of the talk. I think over the last few slides, you saw that Emulate has done an outstanding job with the engineering in really replicating the renal flow that you would see within a physiological system, as well as then the interaction of both the vascular as well as proximal tubule cells. And this was shown to actually result in appropriate membrane polarization, which is critical for the appropriate disposition of the transporters on either the apical or basolateral side of the cells. So moving forward, we wanted to make certain that we were seeing the appropriate expression of the relevant transporters that are of interest to pharmaceutical companies in terms of the research and development, as well as regulatory bodies in terms of both the elimination of molecules as well as then the potential for drug-drug interactions. Make certain that the coordinated membrane disposition is actually translating to the activities that we would expect. And then from there, make certain that we have not only measurable activities from a messenger, but functional activity of the various transporters against target molecules. And then that would translate into the expected inhibition profiles that we would see in order to generate the adequate clinical drug-drug interaction models. So we ask ourselves, why is it important? Why do we need an advanced renal model as opposed to a number of the technologies that we have available to us today? And the simple answer is that we know that compounds will interact with multiple transport systems as they're progressing through the kidney, or in particular here, the proximal tubule. Using metformin as an example here of the interactions with OCT2, as well as then MATE1 and MATE2K. And it's the interaction of various compounds with those transporters that would result in the potential for a drug-drug interaction. Many of the systems that we have today are singly expressing or don't necessarily fully represent the tissue, so it can be difficult to model outcomes when you do see these combination of effects coming through. So one of the major things that we needed to do first and foremost was to ensure that we have appropriately expressed transporters within the system. And what we're showing here is the relevant transcriptomic data looking at both the kidney chip model as well as then those from a transwell system that was generated with the same cells. And what we can see is that the chip model actually maintains the differentiation that you would expect to see as well in a relevant human model with the continued expression of all of the relevant transporters. Yes, in some instances they are upregulated, but the thing that we need to remember is that within the human kidney that this is actually looking across 4 different donors in terms of the data there. So in many instances, this is actually within what we would expect in terms of the standard deviation and taking that forward. And this in and of itself translates very, very well in terms of the activity that we see within the systems. So within the transwell system that we described in the last slide, what we see is that there is continues to be the action of PGP, particularly around use of the active transport of digoxin. And we do see an efflux ratio greater than 2, which is what we would use as our standard in terms of accounting for active efflux. However, when we start looking at the other relevant transporters, OCT2, MATE1, MATE2K, as well as OAT1, we're not really seeing that differentiation come through over time. So the efflux ratio stays right around that 1. We never make it to that level of 2, which would be required in order to show an active model that we would basically use to take forward to say that there is an interaction going there. However, that changes when we move forward to the chip-based model. So again, showing the exact same compounds here, excuse me, digoxin for PGP, metformin for the mates, as well as then OCT2 activity. So when we look at those transporters expressed on the apical membrane, we see a much greater efflux ratio for PGP as compared to the trans-well membrane, as well as then metformin, basically showing the activity that one would expect there. Again, just meeting that threshold of 2 on day 8, but then exceeding it on day 14, showing us good active transport within the system. The same can be said for those transporters that are expressed on the basolateral membrane. So looking at PAH, or the para-aminohipurate, as well as then TEA, tetraethyl ammonium, for OAT1, as well as OCT2. So again, on both day 8 and day 14, we are seeing an efflux ratio greater than 2, and that is maintained throughout the system. Likewise, we do have additional data that's not presented within this presentation for adefovir, cephridine, as well as valsartan, showing the broader range of activity across a number of different molecules. So while activity in and of itself is great, it's really the first step. It's really the initial step that we wanted to accomplish. What this really comes down to, though, is whether or not we're able to show a strong assessment of the drug transporter interactions. And again, we set these experiments up looking at compounds that are mentioned, that are appropriate within the guidelines that are presented by the various regulatory bodies, as well as then looking to set this up in a physiological manner. So the experimental setups, the victim drug was administered to either the apical or the basolateral side of the chip, so that we could basically look at the different efflux ratios there. The inhibitor was infused through both the apical as well as the basolateral channel. This is to replicate what we would see with the inhibitor or the perpetrator that would be present in the bloodstream on the basolateral side, as well as then what could be present on the apical side entering the proximal tubule through just standard GFR. The parent was determined at multiple concentrations of the inhibitor and then plotted in order to determine the IC50. And in this case, because of the concentrations that we're utilizing, the IC50 can be used as a rough marker for the KI within the experiments. So the specific data that I'm showing here is the interaction against PGP for digoxin and quinidine, with digoxin, of course, as the victim drug and quinidine as the perpetrator, as you see here. What we can see is over the course of increasing concentrations of quinidine, we can actually see the efflux, if you will, normalize out, so that in terms of the efflux ratio, we're actually seeing a very strong concentration-dependent inhibition going forward, with an IC50 in this case of 0.723 micromolar that's determined over the course of the experiment. This is then replicated, I should say, or enhanced through the assessment of a number of different drug interactions. So in this case here, our victim TEA, perpetrator cimetidine, as well as then cimetidine against metformin, looking at PAH for probenecid, and then probenecid again against adefovir. And in every instance, again, we can see a very dramatic and very clear-cut concentration-dependent interaction throughout the system, with the IC50s reported there for both PGP, as well as in looking at the OCT1 versus mates, OCT2 in mates, as well as then the organic anion transporters as well, with probenecid acting as the perpetrator. But what this really comes down to is, what is our ability to model the data? So a lot of the inhibition experiments that I was showing previously could frankly be replicated in some of these single transporter expressing models. But where those models tend to fall apart is that when we start trying to develop clearance predictions from a proximal tubule. So we took the data that was generated here, and we generated models to actually look at what those clearance predictions are, and then from there, taking that into a model for drug inhibition, and then the potential changes of AUC. So the upper limit in terms of the modeling experiments was calculated as the summation of the clearance from the glomerular filtration, as well as then the proximal tubule secretion, assuming negligible distal tubular reabsorption. Because as we think about the totality of renal clearance, it is a combination of all of those systems. The proximal tubule secretion clearance was calculated from the tubule kidney chip model and the efflux ratio, assuming active and passive transport equilibrium achieved between the GFR as well as then the proximal tubule blood supply in vivo. Distal tubular reabsorption, that was assessed assuming equilibrium achieved between the unionized compound and the proximal tubule, and the unionized unbound fraction of the proximal tubule blood supply in vivo. So as we look at this, we need to remember that the distal tubular reabsorption is different between the different compounds. So digoxin, while we do see active transport of PGP in the proximal tubule, there is significant reabsorption that occurs within the distal tubule. And that is reflected in the model that we have here, or the actual in vitro predicted clearances versus the actual that are seen in a clinical environment. So not only are we looking then at the kidney chip values, but then also looking and comparing that to the transwell values. And what we can see is that in many times, the proximal tubule kidney chip predicts a clearance value of only 85% or 85% of the clinical value for Defivir, 63% for Metformin, and 72% for PHA. That's as compared to the transwell models, which in many instances are under-predicting that clearance and yielding, if you will, differential results as compared to what one would expect to achieve in a clinical environment. And hence the gap that we see between the transwell with the current models and the clinical trials that are then required as part of the drug interaction guidance documents. So from there, we needed to extrapolate this data and really explore the potential of the model, or the application of the model, to translate and really show that drug-drug interaction and compare that to a clinical setting. So again, the mathematical model set up using single compartment PK elimination model, basically just within the renal, and using inputs of dosing regimen from published clinical trials in terms of the kidney transporter drug-drug interactions, and then utilizing the outputs, comparing the predicted clinical impact to clearance and area under the curve, and comparing that to reported clinical values. So again, focusing on the first example there of the Digoxin-Quinidine model. Again, looking at digoxin, or the victim, basically a single IV dose at 1 milligram. Quinidine is the perpetrator, 200 milligrams orally 4 times a day. And again, the population of healthy volunteers. The clinical trial across 4 different replicates showed an average total clearance decrease of about 46% across the two. And within the kidney trip prediction, we're actually seeing an intrinsic secretion clearance decrease of 67%. However, then when we bring in the totality of their total renal clearance, that matches that which we saw in the clinical state, again of 46%. This in and of itself translated very well to what we would expect to see in terms of the change in AUC as predicted. So with digoxin, given the clearance and the half-life of the compound, we do see in a normal setting complete clearance within a 24-hour period. However, because we're inhibiting that in the presence of quinidine, we do see accumulation within the clinic, and that is modeled very, very well within this experiment here. So, excuse me, the kidney chip prediction basically sees a 59% decrease in digoxin clearance. The clinical study basically showing approximately a 54%. So again, very, very well predicted there. Likewise, too, what it comes down to is the overall change in exposure, seeing 128% increase in digoxin AUC with the kidney chip prediction, compared to 166% increase with digoxin exposure in the clinical trial, something that would not be accounted for or seen within a transwell model. This, in turn, is also replicated through the other transporter interactions that we would expect to see. So as previously talked about with PGP, with quinidine and digoxin, we do the same with cimetidine and metformin, as well as then probenecid and nadefovir. Again, with metformin, we would expect to see a reduction in renal clearance of about 27%. The predicted clinical impact from the renal chip model is a 20 to 38% reduction in renal clearance. Likewise, an AUC increase of metformin in the clinic is observed at approximately 50%, with the chip model, again, being within twofold of the 26 to 60% increase in AUC. When we look at nadefovir as a victim drug, again, we would expect to see there an increase, at least what we saw within the clinical publications, is an increase of AUC of approximately 82%, which again is well modeled with an AUC from the chip model, an increase there of 64% as a result of a 39% overall reduction in renal clearance. So as we look at this chip, it really provides that bridge that we're looking for in terms of the need for an advanced renal model. The current static systems are good for determining whether a compound is a substrate or inhibitor of a transporter. We can determine Ki values, but what they're missing is the interaction of the transport systems. The chip, as we've demonstrated throughout the course of the talk, does express the multiple transporters that one would see within the proximal tubule, and the interaction of those transporters are well modeled in terms of translating the data to clinical outcomes. So we're no longer jumping from a static model that gives us limited in vitro data straight to a clinical trial. There's something that's in between that fills that gap, which is the proximal tubule chip that has been well engineered by Emulate and that we're utilizing at Covance in order to generate relevant data. So again, to summarize, what Emulate has been able to generate is a very well-engineered chip, a very well-engineered system that is easy to use, that emulates the proximal tubule that one would see in a physiological system. Renal flow is replicated, which allows the system to actually behave very similar to what one would see in the physiology of an individual. We do see the interaction between the renal vascular and the renal tubule, endothelial and epithelial cells as those move forward. Likewise, with the appropriate membrane polarization, as well as then the expression of the various transporters, we are seeing the deposition as appropriate to both the apical as well as in the basolateral side of the cells. But most importantly, these are all coming together to give us measurable activity as well as then the appropriate inhibition profiles that we would expect to see. And all of this comes together to be able to translate into relative clinical outcomes that can be utilized to better predict what could be happening within a patient as a compound is moving forward. So with that, I'd like to acknowledge the teams at both Emulate as well as at Covance that are responsible for the generation as well as then the modeling of this data. So Josiah, responsible for the modeling. Anantha, responsible for much of the actual in vitro data that we're seeing, and the teams moving that forward. So with that, we'd be very, very happy to take any questions that you have. And again, thank you very much for taking the time to out of your day to spend with us.
Thank you, Dr. Anderson, Dr. Jang, and Dr. McKenzie for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So, let's get started. Our first question is, how long do epithelial cells remain active? And over a period of time, do they change physiology or biochemical properties?
I will take this question. This is KJ. Yes, based on our snapshot that I presented, we could go usually up to 14 days in culture total. Based on our characterization morphology data, we think that window allow really the multiple different types of functional states and other viability and different application, including toxicity, transporter studies. And over time, what we noticed was morphologically, proliferation rate became a little bit over. So that's the kind of optimal window that we could propose. And we can see a little bit of decline after 14 days in culture.
Wonderful. Thank you so much for that answer, KJ. So we'll move on to our next question, which is, do you see any expression or morphology changes in the 3D kidney model with mechanical stress?
Yeah.
So, go ahead, KJ.
Yeah, I'll take first then. So based on key transporters or key protein expression based on immunofluorescence staining or Western blot to check protein level and gene expression level as well, we could see improvement overall expression level and also correct localization when we compare the chip under the flow versus static condition. Also based on our confocal Z-section image with the apical and basolateral marker staining, we could also notice that increased cell height under the flow in the chip compared to the static culture, which show very flat cell structure.
Thanks, KJ. And if I can add as well, so we also did look at the various experiments initially under flow rates to optimize the flow rate, and we did see, if you will, differential transporter interaction and the ability, if you will, for reabsorption with the flow rate. So that has been optimized within the chip.
Wonderful. Thank you so much for that. So our next question, have you done any site-to-site comparison for this model? If yes, how was the variability?
Yeah, I will provide the answer first. So we did a comparison study, especially between Emulate versus Covance site. Last year, when we were ready to transfer the technology, we measured some of the substrate that we presented today based on some efflux ratio that we could see, which was measured by LCMS from Emulate site, and then radio-label substrate from Covance side. We could see tight variability. It was within 10 or 20% variability initially. And then from there, what Covance team did was further really tweaking, optimizing the condition to improve even better overall efflux ratio and functionality. So based on initial comparison, we could see quite a tight variability between site to site.
Wonderful. Thank you so much. So our next question is antibody effect, could that be monitored using this Emulate chip?
Yeah, so this is Don. I'll take that one. So yeah, so with the push towards large molecules within the industry right now, we do believe that this model could be looked at to gauge the interaction of antibodies on the basolateral side, so the antibody in circulation, the other advantage is not only would we be able to look at just a straight antibody, but we could also apply it to other types of therapies like antibody drug conjugates, nucleotide therapies, and the like. And for instance, with an ADC, we'd be able to look at the antibody itself, the small molecule warhead by itself, as well as then the conjugate.
Great. We want to remind our audience that any questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speakers via the email address that you provided at the time of registration. So let's move on to our next one. If these, has this data been published, and if yes, is there any references for more detailed reading?
Listen, I'll take that one as well. So the manuscript is currently in progress in terms of coming together. The plan was for us to initially present this at ISSX as well as then with the webinar, but we do expect the publication to be coming out in the near future.
Thank you so much for that clarification. So our next question, have you tested different flow rates in the kidney chip? Or excuse me, what types of additional applications can we do using the kidney proximal tubule chip?
So, Mr. Donegan, there are a number of different things that we're looking at, particularly with the second horizon of the project beyond just the drug-drug interactions. But taking it back to that is the potential to use this to assess nephrotoxicity based on the drug-drug interaction, where, for example, you could see active uptake on the basolateral side, but then prevent the efflux into the apical side, causing a concentration effect within the proximal tubule cells themselves, resulting in nephrotoxicity. I also know that our colleagues at Emulate are also looking at a couple applications. So, KJ, if you want to.
Yeah, sure. Yeah. So additionally, we can do also disease modeling. We can kind of induce injury status, for example, treating TGF-beta, for example. And then we can also utilize patient-derived cells in the chip as well to model disease, certain disease, including acute kidney injury models. And then we can do some therapeutic testing for efficacy perspective. Also, we can do COVID-19 related research as well. There are multiple cell types expressing AC2, for example, including podocyte, mesenchymal cell, and proximal tubule cell as well. So kidney cell highly affected by COVID-19, causing acute kidney injury. So those type of applications are also available.
Thank you so much for the clarification from both of you. So our next question is, there's a couple parts to this next question. So the first part, does the inhibition of Digoxin-Quinidine in the model suggest the DDI occurred in the proximal tubule? Now, do we have clinical or preclinical evidence to support this? And the last part of the question, can the model used for predicting digoxin absorption DDI as an alternative of CACO2 CACO2?
Yep. So thanks, Alexis. So in this case, we are showing, if you will, the inhibition of digoxin, or PGP, might be a better way to put it, in the proximal tubule, and then using the computational model for the overall impact from the distal tubule. In terms of a substitute for CACO2, I would argue that we're looking at a very different system with the intestinal. And likewise, the current hypothesis, current theory is that PGP efflux really doesn't impact overall absorption in a significant manner. But with that said, we also do know that there is an intestinal chip under development, and some technology, if you will, linking up the organ system to better emulate, if you will, what's going on with that system is better than trying to substitute one organ system for another. KJ, I don't know if you have anything else you wanted to add.
That was good. Thank you, Don.
Wonderful. So we still have time for a couple more questions, but again, as a reminder, any questions that we don't get to today or those submitted during the on-demand period will be addressed by the speakers via the contact information that you provided at the time of registration. So our next question is, let's look for one. Okay, how about this one? Was 60 microliters an hour the optimized flow rate?
Yeah, I can take this question. The answer is yes. We tested different flow rate, including 60. And then when we tested different flow rate, what we found very interesting was compared to static, we could see clear differences in terms of transcriptomic global gene expression profile, also functionality. However, among different flow rate groups, we didn't see that major differences. So based on transcriptomic expression data, also functionality data, efflux ratio, we haven't seen any big major differences. So 60 microliter was based on that testing optimized flow rate that we are applying.
Thank you, KJ, for that answer. So our next one, in terms of drug screening, what is the rate of throughput?
This is Don. I'll take this one. So this model right now really is not designed for early screening. The theory and the way that we're looking at it is the singly expressed models do a very good job and are a good initial start, if you will, in terms of the experimental processes for compounds moving through the system. Where we see this model really fitting in is that gap between, early screens or the singly expressed systems and clinical trial.
Wonderful. Thank you, Don, for that answer. Okay, so we'll move on to our next question. We've gotten so many good questions, and thank you so much to the audience for all of these questions. Let's look for our next one. Okay. Here's one. There's a small variation in the clinical comparison with the chip. Is it within the error limits of the measurements, or do we need further improvement in the model?
Yep, so this is Don. So we would always be looking to improve upon the model itself, but in this case, in terms of the variation that we see, we do believe that's within the biology of the system. And what we would expect to see within just the natural laboratory experimental error that you would have in any sort of biological system and testing.
Thank you so much, John. Now our next question. Do you have any data on megalin or cubulin?
I'll take this question. Yes, we have the data, both expression and functionality. In terms of functionality, we measured albumin uptake, and we could see increased albumin uptake in the chip compared to static culture. Also, when we correlated the data from the chip to in vivo, it was within physiological relevant range.
Thank you, KJ. So our next one, and I think this one is for you as well. Do you plan to create a kidney disease relevant models on the chip?
Yes, it is possible to build different types of disease models for kidney. As I briefly mentioned, there are different ways making disease states for the renal model. One is utilizing patient-derived cell. So one example we did was creating a diabetic kidney model by utilizing the cell proximal tubular cells from type 2 diabetes patient. So that was one proof of concept type of study that we did. We could see maintenance of phenotype of that patient maintained in the chip over time. And when we tested therapeutics, we could see efficacy as well. So same approach can go with other disease model type as well. So if you maybe leave your contact information, we can provide additional detail.
Thank you, KJ, for that. Okay, so moving on to our next question. What about, okay, let's go to, okay, here's one for you. Can we use organoids for the kidney chip model?
I can take the question as well. Yes, we can do that. So far, we have been utilizing organoids for multiple different organ models, including intestine, and it is possible for other organ models, including kidney. So the way we've been doing is utilizing organoids, and we expand them, and then before putting the organoid in the chip, we fragment them in smaller size. So we create a monoculture, monolayer first, and then we differentiate them in the chip under flow and additional mechanical forces. So the benefit still culturing organoid in the chip is having vesicular compartment, allowing really additional really cellular interaction between different cell types. So similar approach can be done for the kidney. And the other component that we could a little bit optimize or test differently is putting different sizes or different methods of fragmentation when we add organoid in the chip. And we can also utilize the top channel by adding different types of or thickness of the gel. So yeah, it is possible with different many different ways.
Wonderful. Thank you so much. So now our next question. In the future, would you develop chips that incorporate proximal tubule cells from different populations? For example, different ethnicities or male or female?
So this is Don, and that's a great question because of the fact that we do know not only within the ethnicities, but then also age-related, and we could even look at the potential of cells from diseased patients. So yes, because this is a cell culture model, we can, as long as we can obtain the cells and make certain that they are well characterized and that the system itself is well characterized, we could basically introduce those to be able to populate the kidney side or the kidney channel, I should say, with various models. But the biggest thing there is making certain that the system is well characterized so that we have faith in the results that are coming forward.
Thank you, Don. Now, our next, looks like we've got time for maybe one or two more questions. So our next one is, can you seed the chips with different cell types, like primary culture proximal tubules?
Yes, I will take this question. Yes, we can do that. The current proximal tubule kidney chip model we have is based on primary cell sources. And co-culture, both having endo and epi, both are from primary sources of healthy human. So that's baseline of the model. In case we have other sources of the cells available, we can certainly use other cell types as well.
Wonderful. Thank you so much, KJ. So it looks like we've got time for one more question. Let's go ahead and look for our last question. We want to thank the audience again for these amazing questions that have come in today. Okay, our last question. Where do you source your epithelial and endothelial cells from?
Yes, I will take this question. It is commercially available cell sources, both renal epithelial and endothelial cells, and it's part of our product, Kidney Bio Kit. So we provide a qualified QC cell sources, both epithelium and endothelium from commercial sources.
Wonderful, KJ. Thank you so much for that. And we want to thank Dr. Anderson. Thank you again to Dr. Anderson, Dr. Jang, and Dr. McKenzie. Do any of you have any final comments for our audience?
Again, I'd like to thank the audience for attending. Some great questions coming through here, and those that we weren't able to get to live, we'll definitely follow up with email answers and so on. And again, looking forward to developing partnerships and taking this model forward to help basically bring new compounds to the market and address many of the unmet medical needs that are out there. Patients are waiting, and we can't slow down the discovery process.
I second that. Thank you so much, everyone, for your time and great questions and interaction. Thank you.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information that you provided at the time of registration. We would like to once again thank you, Dr. Anderson, Dr. Jang, and Dr. McKenzie for their time today and their important research. We would also like to thank Labrits and our sponsor, Emulet, for underwriting today's educational webcast. You can view the webinar on demand. Labrits will alert you via email when it's available for replay. That's all for now. Thank you so much for joining us, and until next time, bye-bye, everyone.